Hi everyone, my name is Sumaer Kamboj, and I am here to present to you on leveraging amyloidogenic properties of alpha-synuclein for its purification in E. coli. So I work in an interdisciplinary lab here at Colorado School of Mines, and a main part of what we do in our lab is having to communicate information across disciplines, and this is very challenging at times. So I made this original PowerPoint presentation to try to explain it very much in depth. And due to time constraints, I might be going a little fast. So I wanted to just give you a quick outline of what I want to tell you about. It's what is alpha-synuclein, the medical significance of alpha-synuclein, and then we'll move to the body of the presentation, which will have what exactly I did, from inserting the genes of alpha-synuclein to purifying it, visualization in 3D, and then confirmation of alpha-synuclein, it being produced, and then final and conclusion. So what is alpha-synuclein? So alpha-synuclein is a protein that is found mostly within the neurons of your body. It is intrinsically disordered, which is a very unique thing for a protein to be. A typical protein has a very distinct structure and function relationship. A good example of this is shown in figure 2 here, which is hemoglobin. Hemoglobin is also a protein that is made of 4 subunits. All 4 subunits have to come together, which are originally expressed in a nice little string, fold, and then all 4 will come and allow an iron to bind to carry oxygen throughout your body. This folding process is very important to the function of the protein. Without it, the hemoglobin is completely useless. So someone might have a near perfect sequence in their DNA for hemoglobin, but if you have one change in that sequence, some 1 point mutation, you can drastically affect how the protein folds and hence affect its function. This is why single point mutations can be so damaging to many cells. But an important thing to know about alpha-synuclein is that it is a protein that does not have a ordered state that is well defined. There may be one that we just have not been able to pin down, but this is not something that's been well studied. So the next I want to talk about is what is the medical significance of alpha-synuclein. And the main 2 diseases alpha-synuclein is involved in is Parkinson's and Alzheimer's. In these 2 diseases, there have been found what are called Lewy bodies. And Lewy bodies are these dense aggregates of alpha-synuclein that cannot be cleared by the cell, and this leads to cell death. Particularly in Parkinson's, this leads to cell death in the area known as the substantia nigra. So the substantia nigra sits at the top of your brainstem, about back from your eye, out to your ear, let's say, and it helps produce dopamine that regulates the way you move. So the cells are dying in this area and it no longer can control the way you move. This results in a nice tremor that you've probably seen with a lot of Parkinson's patients. Furthermore, alpha-synuclein, when it forms the structure, there have been many reported studies on how it can do it, but what's most interesting to me is that this protein can act as what's known as a prion. And if you're familiar with biology, a prion is a very scary thing. This can be cause diseases such as mad cow disease, Creutzfeldt-Jakob, and even Kuru in some extreme cases in Papua New Guinea due to cannibalism. So this is a prion is a misfolded protein that when it's introduced to other proteins, it causes other ones to misfold. So once you have one improperly folded alpha-synuclein, it will start stacking with others and lead to a full aggregate, which will then lead to cell death. Now this is all theorized. There's not a solid body of evidence to support this. There are some people that report that alpha-synuclein aggregates are not essential to Parkinson's disease, but may just be a product of it. But my research here is how to produce alpha-synuclein. So being able to produce more of it will help research further in this direction and be able to maybe enlighten more pathology of alpha-synuclein. So now we've covered the background. Let's move into the body and show you exactly what I've done for the past year. So I'm trying to produce alpha-synuclein, which is a human protein, in E. coli. And to do this, you need to know about something called recombinant DNA technology. This figure 6 here that I've taken is coming from Khan Academy. I highly recommend looking at their videos if you want to understand recombinant DNA technology better. But basically what recombinant DNA technology is using another organism to express a protein. You're kind of splicing the DNA together. You're recombining it. So I've done this with E. coli. And shown here on the right in figure 6, actually, sorry, now, we are showing that I have properly expressed the protein. So this is a protocol known as SDS-Page, which allows you to separate proteins by size. So we have a nice ladder over here, which is of a standard size, like a laser pointer, nice ladder that these are known sizes of proteins put in. I put in my 2 samples, and out I get this nice thick band of alpha-synuclein. Both of these cells had the capability of making alpha-synuclein, but only in this one, IPTG positive cell, did I actually induce the expression of alpha-synuclein and allow it to be made. And this test and analysis through SDS-Page allowed me to show that alpha-synuclein is actually being produced. So next, I'll talk about what the purification protocol looks like. So something that's very unique about this purification protocol is how quick it is and how much you can make. I wanted to list out exactly how this is done, so I even gave like a quick step-by-step protocol. If you want a more detailed one, be looking in scientific reports for our publication, and you'll have all the materials and methods you need to do this yourself. So starting at day 0, suppose my advisor tells me we need some more alpha-synuclein. I'll go to the freezer and take our little glycerol stocks, which contains E. coli with the DNA put in it, and I'll make it more of it. I'll grow a bigger stock. The next day, I'll come in and I will have a nice seed stock, I call it, and I'll use that to grow in even a larger amount of media. And this grows to our size in our 500 milliliter shake flasks here. Just a quick disclaimer, these images are not the exact shake flasks, the exact protein that I was producing. This is a different one, but this is just a good visual. And this is growing E. coli. In one of these flasks, I will add the inducing agent of IPTG, and this will induce expression of our protein. And then our cells will then be used to keep pumping out alpha-synuclein. Further from there, I'll take this broth, centrifuge it down, and pellet it to form a solid E. coli pellet. That pellet will then be processed by lysing the cells. Lysing literally just means to break open, so it'll take the protein from inside, bring it into solution. I will then centrifuge to remove that excess cell debris, leaving the soluble protein in the supernatant, and then I will filter to purify alpha-synuclein. Now what's really interesting is what I've been seeing in these last few steps of hour 7 and hour 8. So in these steps, we saw something very unique to alpha-synuclein, and that is the formation of aggregates. Typically after centrifugation, after lysis and then centrifugation, you get a solid cell pellet at the bottom that is full of cell debris. But no matter how much I centrifuge, no matter what speed, no matter what time, I would not get it to precipitate down. I formed this kind of viscous mess which refused to settle, as you can see here in figure A, in part A of figure 10. These were then looked at under a microscope to see that they have a very unique shape. They're kind of crystalline, kind of nice rigid edges. This started to hint that maybe we're forming something here that might be like an amyloid. Further confirmation showed that purifying it in this way, we get a dense aggregate up here and then a nice clear filtrate. Up here, all of our aggregates are staying, but when I look down in this sample here, if you look at the SDS-Page again, I have a nice clean band. This section here is without purification, and this is after. I have removed all of these excess proteins and can confirm using Western blotting, which is an antibody-based method, that here is alpha-synuclein. So this kind of a result is so clean that it's almost surprising. This shouldn't be there. This was almost too easy. So we started looking into what these aggregates could be. And what we found was that these aggregates could actually be alpha-synuclein acting as a glue to remove the native E. coli proteins. This glue-like mechanism of alpha-synuclein is kind of known as it causes cell death, but having it occur in E. coli to assist in purification was something very new to us. So here we have really shown a new method for purifying alpha-synuclein, and in the process we have created what could be used as a Louis bottle model of this amyloid structure. And that is kind of a very important kind of statement. So let's talk about the significance of that. What's really unique about the protocol is that we can produce alpha-synuclein in 2 days. That is something that is not really done anywhere else in the world. Usually you have 4 days dedicated just to purification itself, which involves a series of pH changes and then chromatography columns. But a 2-day purification from someone telling me I need alpha-synuclein to me giving it to them in their hand is a fast turnaround. This kind of a fast turnaround should accelerate research around the world and maybe start providing a new model for Lewy body formation. studying this further can really help understand how Parkinson's develops in the body and what the roles of different proteins are in neuronal cells. So that's everything I have to do for you today. It's been quite fast. I know we don't have much time, but please do email me if you have any questions, or you can also reach out to Dr. Sumaer if you have more logistical questions. Thank you for your time.